Thank you all for coming. I'm honestly surprised at the turnout. This is so great. Thank you so much. um, for those of you who haven't met me, because I'm still relatively new, I haven't even been here 2 months. My name is Bridget Shell-ebrat, and I am the new genomics scientist. I work in zoology and health sciences, and I'm here today to talk about mollusks. Yay! So, if any of you saw my talk back in February, I think I spent maybe 30 seconds talking about my PhD work. I think it was one slide. So now we're going to stretch that out and look at not only the snails that I studied in my PhD, but also the phylum of mollusks. In this talk, which is Shell-ebrating Symbiosis-Microbial Partnerships across the phylum Mollusca. So first, I just want to begin by generally talking about biodiversity. Biodiversity can be defined as the variety of life, of genetic material, of functional traits, and it's essential for long-term ecosystem health and stability. And when we think of biodiversity, we often think of this kind of image here, all of these individual species which together make a biodiverse world. And research on biodiversity loss often concentrates on these impacts on these individual species. However, there are important aspects of biodiversity, which are the complex biological interactions between species, or their symbioses. And one such example is between a host and its microbes. So, a microbiome is a community of tiny living organisms like these microbes, like bacteria, viruses, archaea, fungi, that live in a particular environment. So, microbiomes can be found kind of all over the planet. They can be found in soil, in water. They can be found in and on our bodies and the bodies of other animals. And microbiomes have come under pretty considerable scrutiny in recent years due to advancements in new technologies. These are really cost-effective, and they help us characterize these bacteria that normally you'd have to culture in a lab, and now we don't have to do that anymore. So this recent surge in research has revealed that microbiomes can profoundly influence their hosts' health. And microbiomes influence things like immune functioning, like digestion, metabolism, development, reproduction, and behavior. And so they can directly impact host health and fitness, which is basically how well an organism is able to survive and reproduce in its environment. So far, microbiome research has focused on only a few main hosts. So the first one is obviously the human microbiome. We've been really intensely studying human health in relation to the microbiomes in various parts of the body, things like the gut microbiome, the nasal, the oral microbiome, things like that. Another big field are human analogs. These are things like mice and rats, where researchers experiment on these model organisms to test their theories related to human health. And then another one is commercially important species. And what I mean by that are these animals that are typically important for agriculture or the food industry. These are things like cows, pigs, chickens. But microbiomes also have significance in affecting these lesser studied hosts, which are wild animal populations. And microbiomes can profoundly influence wild animal health. So for example, in these corals here, corals have been a primary focus for microbiome studies because they have a lot of associated microbes, which can be specific to a species. They really help it out. They help with nutrient cycling, pathogen defense, and they can be lost through the process of what we know as coral bleaching. And then the disruption of microbiomes also poses a serious threat to a lot of wildlife populations. So for instance, in these howler monkeys, gut microbiomes can be altered in negative ways due to the level of fragmentation caused by human development in their habitats. And also microbiomes can help their host species in camouflaging and avoiding predation, like in this Hawaiian bobtail squid. However, even though I just mentioned that squid, the examples of known molluscan host and microbiome symbiosis are the exception rather than the norm. It's not very widely studied. So while we do know a lot about a certain few mollusks, like that squid, the phylum is really broad and it's really expansive, and there's a lot left to learn. So today, I'd like to share some more about mollusks and their microbes, and investigate several aspects of the microbiome of one particular mollusk that resides right here in the Colorado Front Range. So first, I'll talk about examples of the various symbioses that we know about in this phylum. And then I'm going to narrow in on a native Colorado snail and how its gut microbiome is impacted by its life history. So first, let's talk about mollusks and their microbes. So what are mollusks? They are a really fascinating group of animals. If we're talking specifically, they are a phylum under the kingdom Animalia. So they are the second largest phylum of invertebrates, I think after arthropods. Arthropods. And they are highly diverse, not just in their size, in their anatomy, but also in their behavior and the habitats that they can inhabit, like deep sea trenches or mountaintops. And this phylum has many orders within it. So these include things like cephalopods. These are things like squids and octopuses and cuttlefish. There's also gastropods, which are things like snails and slugs. There's bivalves, clams, scallops, oysters. There's polyplacophora, which are chitons, among other things. And a lot of mollusks are threatened. About 42% of the recorded extinctions since the year 1500 have been mollusks, and most of them have been non-marine mollusks, so terrestrial mollusks. And there's some contention about what makes a mollusk, but I'll point out some features that are pretty universal. So the first is they have this body that's consisting of a solid muscle. They have this mantle, which has a cavity that's used for breathing and excretion. They have a radula, which is sort of like a tongue-like rasping device. I'll talk more about that later. And then a lot of them have a very similar structure of their nervous system. And mollusks, again, they have a huge amount of biodiversity even within them. They range in size from the very large, like this African land snail, to the very small, like this microsnail. They can be really colorful and beautiful, like this is the blue-ringed octopus, and this is the variable neon nudibranch, and that serves as a warning for their toxicity for potential predators. Some examples from other orders are, for example, there's this scallop, and if you see these blue dots all along its mantle ridge, those are all of its eyes. And this is a chiton, so if you've ever been like tide pooling, you've probably seen a chiton before on the rocks. Mollusks also include just some of the cutest things you've ever seen. And this is a sea slug. It's called the leaf sheep. I think you can kind of tell why. And this actually has a symbiotic relationship with algae, which helps it perform photosynthesis, which is really cool. Mollusks also include some of the most horrifying things you've ever seen. This is a big fin squid. I don't think anyone's ever seen this in person. Correct me if I'm wrong, but they can live in really deep ocean trenches. These arms can be 26 feet long. So I hate that. And anyway, if you'd like to see kind of nicer mollusks up close, you should stick around after this because they have some of the education collections for you to touch. Okay. So let's talk about some examples about how we know microbes help mollusks out. So the microbiome can assist mollusks in these kind of predator-prey interactions. As I mentioned earlier, there's this Hawaiian bobtail squid. This is a really tiny nocturnal squid. It lives in shallow coastal waters. It hides in the sand during the day, and then at night it comes out to feed. And this has, you can kind of see it here, it has this bioluminescent glow, which is that blue glow right at the bottom of its mantle. That comes from its light organ, and it can manipulate the glow in its light organ to match the intensity of the moon and the stars, and it basically masks its silhouette. And so this is called counter-illumination, and this helps it conceal itself from any predators that are beneath it, but also from its own prey that it's trying to hunt. And when these squids are born, they're completely sterile. They have no bacteria. Within hours, they're colonized by this one particular kind of bacteria. We don't know how it finds it, but it becomes colonized by this bacteria, which is called Vibrio fischeri. And as you can see, this is the bacteria under regular light, and this is it glowing. So that is what gives it its blue bioluminescence, which is really cool. You may have heard of cone snails. Cone snails are venomous marine snails. They have this really deadly neurotoxin that they keep in harpoons, and they use them to immobilize their prey and also defend themselves against predators. And studies show that they also have a microbiome, and that they contain these diverse bacteria that produce metabolites which exhibit neurological activity. And we think that that aids in creating this really potent neurotoxin inside of them. Nutrition and diet and metabolism are all greatly aided by microbes in a lot of mollusks. So it's kind of hard to see here, but this is a bone-eating snail. It has microbes in its gut to digest its very specific diet. Any guesses what the bone-eating snail eats? It's bones. So they exist at these whale fall sites. And a whale fall is just when a whale dies, it sinks to the bottom of the ocean. It provides this kind of whole ecosystem. And these snails are some of the things that help cycle these nutrients back into the ecosystem. They eat the whales. There are also a lot of species of bivalves that are chemosynthetic, and that means that they can make their own energy from chemicals. And they can use microbes that are found in their gills to do that. Like this is a clam that lives in a deep sea hydrothermal vent. And then there are some grazing snails that could use bacteria as a food source itself. So as Hawaiian tree snails become increasingly endangered or extinct in the wild, research is really rushing to figure out what microbes are essential to their survival, especially in their guts, because of their very specific nutritional needs. And many snails scrape their food sources with their radula, which is one of those features I mentioned earlier. And a radula, this is a very zoomed in version of it. This is under a scanning electron microscope. Usually it's the size of like a hair if you look at it in person, at least for these snails. So snails can farm bacteria using these radula. This is a ribbon, basically, of chitinous teeth. This is all teeth. These radulas are plastic in nature because they're kind of like sharks. They're always producing more teeth. So they can adapt to different feeding substrates. There's a little bit closer up, and then there's the teeth really close up. So these radulas, these teeth, they can change shape. They can adapt to different substrates, which allows their substrates to support diverse microbiomes themselves. That can foster the growth of specific microbes that the gastropods can then graze on. Okay, so I've talked about a variety of mollusks, and we've seen that they have a proven and extensive symbiosis with a lot of different microbes. They exist in a lot of different places, deep sea vents, Hawaiian forests, dead whales, and we know that the gut microbiome, in particular, is also important in assisting nutrition and digestion in many mollusks. So when I was doing my PhD, I wanted to further investigate the gut microbiome of an unresearched mollusk. So I needed a species that was easily accessible, something that was abundant, something that was easy to manipulate, and something that we already knew some life history and ecology about. So with this in mind, I thought about, is there an important Coloradan mollusk whose microbiome has not yet been studied? Something that lives close by, even in the Front Range, that we can learn more about the gut microbiome from. And there is, yay. So one molluscan species that lives all around us here in Colorado is called the Rocky Mountain snail, appropriate. But despite its extensive presence across the Coloradan Front Range, it's a species whose microbiome it had never been studied before, not even in the genus. So here I'm going to talk about some of the work I did during my PhD, and all of this is focusing on this snail, the Rocky Mountain snail, and its gut microbiome. So, the Rocky Mountain snail, its scientific name is Oreohelix strigosa. I just say that because it's fun. Is a land snail. It's native to Colorado. It's also native to the western United States, all along the Rocky Mountains, basically. These snails are detritivores. They have this kind of unique diet. So being a detritivore means that you feed on decaying things, decaying wood, decaying vegetation, things like that. They have what's called very low vigility. And vigility is just low dispersal. So these populations don't disperse very far. They only disperse maybe a few meters in their whole lifetime. Imagine if you were confined to just a few meters in your whole lifetime. It's crazy. So they're restricted to these kind of smaller microhabitats, and we call this like a sky island type of distribution. So if you've heard of an island distribution, it's surrounded by water, but instead, since they're limited to these moister areas that are high in elevation, like mountaintops, they have like a sky island distribution. They're surrounded by air. And importantly, I should tell you, I did my PhD at the University of Colorado Boulder, and I also worked in the CU Museum of Natural History. And yes, go Buffs. And that museum collection, the founder of that museum, Junius Henderson, was a big fan of these snails. And so he collected a lot of them, as you can see. He collected a lot of them around Boulder in the Front Range, and that's kind of what started the invertebrate collections. So we have this really prolific presence in the CU Museum of these snails from the 1920s to the present day. So we can use these museum records to find existing populations. It makes it a lot easier. But so far, no published studies had characterized any microbiome in Oreohelix, as I said, even though it offers a lot of benefits in working as a model species. Another interesting thing about these snails is that they're ovoviviparous, and all of that just means that they give live birth. And that's kind of unique in snails. A lot of them lay eggs, which later hatch, but these, they just pop out baby snails that look exactly like them. So that's an example of a parent and a juvenile. So this gives us a really unique opportunity. You can directly compare the baby to the parent. And often microbes are passed from parent to offspring. You probably got some microbes from your mom when you were born. So this is really cool. So again, nobody had studied these snails' microbiomes. So before you can study a microbiome, you kind of have to establish that there even is one. So while, as I said, we're all interacting with bacteria in some way, not everybody actually needs a core microbiome. Not everybody needs these bacteria that are always there in their bodies. That's a common misconception. I see that in papers all the time that says all animals have a microbiome, and that's just not true. So there's things like caterpillars. They only have what we call transient microbes. And that just kind of means they're passing through. They don't live there. They get picked up by the host through maybe their diet, maybe their environment. They work their way through their body, and then they exit. So we've thought that we could use aspects of this Rocky Mountain snail's life history, like knowing their diet, knowing that they give live birth, to help determine which, if any, microbes are consistently found in their guts. So to check if a species has a core microbiome, it's useful to compare different groups and see if microbes persist in all of them. So since we know that animals can take up microbes in their guts from their diet, you can, for example, take away their diet and see what happens. We also know that these snails give live birth, so we can check if baby snails are born sterile or if they already have microbes inside of them from their parent. And then we can compare these groups with just kind of normal wild adult populations and see if there are common microbes. And spoiler alert, there are. So when we compare all three groups, we found that there are certain bacteria that are found in all of their guts. That supports the idea that there is a core microbiome. So that's helpful information to know. And then using that, we can look at microbiomes across certain environmental gradients to see how they might be changing. So to do this, you need to go out in the field and actually collect snails. So you could go out and find these snails yourselves in many parts of the Colorado Front Range. If you're looking to have a good time hiking very slowly and staring at the ground. So I went quite a few places during my PhD. I went to, this is the Rocky Mountain Biological Lab. This is in Gothic, Colorado, kind of near Crested Butte. A lot of ski towns during the summer. Really, snails love a ski town. Vail, Frisco, Steamboat Springs. This is Hanging Lake, which is near Glenwood Springs, and I got to go there, and there was a huge population of snails here. But that was in 2019, before the fires of 2020. I haven't been back, so I have no idea if they're still there, but if you go to Hanging Lake this summer, let me know if the snails are okay. So if you want to find them, they're most active in the summer months, starting right about now to maybe September. They come out right after it rains. They really love aspen groves. And once you find one, you'll probably find a lot because they don't disperse very far. But what if you don't want to drive and hike all around Colorado and the Rocky Mountains? That is the power of museums. So we can sample wide ranges of organisms, not just by their geography, but also the time that they existed in. And museums are basically time machines, right? They allow you to visit whatever time and place that you want. But at this point, microbiome research hadn't really realized the utility and the power of museums. Microbiome data from museum specimens, it wasn't clear that you could get it from things that were preserved decades or a century ago. The longest study up until this point was maybe 10 years, things that were 10 years old. We know that, especially in the CU Museum, we had things going back to the 1920s, so a full 100 years. So I wanted to see if we could use these long-term preserved snails for microbiome research. So what I did was I went into our CU invertebrate zoology collections. I took snails that were preserved at different time points, some that were preserved in the 1920s, the 70s, 80s, 2000s, 2010s, and I just compared their microbiomes. So in short, the long-term preservation, being preserved in alcohol for a long time, actually only impacts the makeup of the microbiome very marginally. About 2% of the variation that you see between these microbiomes can be explained by long-term preservation. And when you look at things, even like just the number of bacteria that are in there, which we call the richness isn't significantly impacted by the time under preservation either. However, we were seeing that they weren't all identical. There were still changes between these gut microbiomes. So if collection time isn't important, what is driving these microbiome changes? Location. So the actual location where these snails were collected explained a lot more variation. This explained 12% in the microbiome composition, and a lot more than just the year that it was collected. So since location encompasses a lot of information, it's possible that this pattern is driven by other environmental factors that can co-vary with location. So these are things like habitat complexity, drought level, diet, disturbances, things like that. And in these museum specimens, there was one environmental factor that we were able to look at across everything from the 1920s to the present, and that's the Palmer Drought Index. That's basically just a measure of temperature and precipitation. And maybe unsurprisingly to everybody here, the later years sampled are much drier than these earlier years. They're much moister. And while we didn't see huge changes in the microbiome composition, we did see differential abundances, I guess, of certain bacteria. So for example, in these later years that are drier, we saw that there were bacteria that have been shown to be associated with drought-tolerant plants or with extreme drastic environmental changes. So we think that the snails maybe were ingesting these bacteria from their environment, maybe helping them become more drought tolerant. So overall, location, environment, geography was so compelling in this study. I decided to keep looking into environmental factors that can vary by location and also expand our area of interest from beyond just Colorado to the whole range of this snail. Forgot about that. Okay. In order to conduct the study, since the CU Museum was really concentrated on Colorado, I drove all over here. No, I didn't. I just collaborated with other museums. And that's also what's so great about museums, is if you don't have what you need, somebody else probably does. So I collaborated with the Florida Museum and the Santa Barbara Museum of Natural History. I borrowed some of their Rocky Mountain snail specimens. And so that increased locations and states that we were able to sample to include not just Colorado, but also Idaho, Montana, Wyoming, Nevada, New Mexico. I also did go back into the field and I collected more snails. By collecting in person, you can gather a lot more information on these sites than what might be typically available in a museum database. And that can help us determine what factors within location might drive microbial changes. So because we went and collected in person, we could collect things about, information about like what kind of vegetation is there, the habitat type, the complexity, the proximity to people, things like that. And here are some examples of just some of those Colorado sites. As you can see, there's a lot of variation even within Colorado sites. So these snails can exist kind of in these open rocky cliffs or in these lush forests. So in answer to the question of if broad-scale location influences gut microbiomes, we again saw the same trend. So location still, even across the whole native range, explains a lot of the variation across microbiomes in these snails. To look at what factors within location might contribute to these changes, we can narrow in on those Colorado sites where we have more information. And we found that many factors within the site that the snail was collected contribute to microbiome diversity. These include things like environmental sources of bacteria. So you can look at the bacteria that are present in the vegetation and the soil, compare those with the snails, and see that they're picking some up. Habitat type was another thing that significantly contributed to microbiome makeup. So again, was it a rocky cliff? Was it a lush forest? Habitat complexity, that is kind of like how many types of vegetation are there? Is there a water body? What's the topography? That had a significant influence on microbiomes. And also there was some significant impact of human impact. So how close were these populations to human development, for example? So, okay, I'm out of time. Okay, so what I hope that you take away from this talk is that microbiomes are hugely important for invertebrates and especially molluscan species. Museum specimens provide a unique and often underappreciated means of gathering ecological information dating from the present to up to hundreds of years ago. And then also these specimens can be used to conduct really wide-ranging geographic studies without the need for you to actually go out and extensively field sample. Some of the implications for this work are pretty wide-reaching. Museum specimens or their preserved parts like tissue samples or DNA, these are vital tools for conducting new and innovative research. We can use museum specimens to understand the long-term impacts of climate and environment on host microbe interactions in communities in the past or in the future. And finally, it's important for researchers to consider adding their specimens into a museum now, for example, adding into the DMNS's collection of preserved tissues and DNA in the cryo facility, because one day future researchers might take those samples and they might use them to investigate new questions with new technologies in ways that we can't yet imagine, which I think is really exciting. So with that, this is everybody who helped me during my PhD, roughly. I'd like to thank all of these collaborators, all these funding sources. I, again, encourage you to come up after this talk and take a look at the education collections. But I'm happy to take any questions that you have right now in the meantime. Thank you. Cube.
Questions?
What do these Rocky Mountain snails do in the winter?
I'm so glad you asked. They estivate. So estivation is kind of like hibernation, where they go into this reduced kind of metabolic state. They hunker themselves down, they dig down into the soil, they bury themselves in there, and they just kind of wait it out. So they persist. I don't think we actually know how long they can live, but they can live multiple seasons, and they survive winters that way. So if you ever wanted to find them in a hurry, I guess you could just dig up the land, and you could probably find them, yeah.
And then I was wondering about, it's going to be a short time period that they have doing things, but would you expect like seasonal variation or monthly variation based on like what's available for diets?
Yeah, so actually, I'm not sure about these snails, but it has been shown that when they go into estivation, they kind of expel all of their gut contents. So every season they're starting over from scratch, which is really interesting for us because then they are really dependent on the year and the season and all of these aspects of the environment. So yes, I think they are kind of starting from scratch and they're kind of recruiting as they go every season. Does that answer your question? Okay.
How
how do you go about like methodologically sampling the gut microbiomes of these snails?
Yeah, so if you are taking them from a museum collection, you go into the collection. We only used ethanol preserved samples, and that's important because we know that if something was under preservation in like formaldehyde or formalin, that really degrades the DNA, and you can't use that anymore, not reliably. So everything that we used in the CU collection had been under ethanol preservation the whole time, and everything that we borrowed, we made sure that it was under ethanol preservation too. So you actually dissect them out of their shells. You kind of pull them by their foot, take out their whole body. The gut is pretty obvious. I wish I had included a picture here, but I thought that might be kind of gross. So it's a darker color. It's like a different consistency. You can tell that it's gut tissue. We only need a very tiny amount of it to actually get extracted DNA and get information about the bacteria. It's the same process in a new snail, except you have to sacrifice it first, which is not so fun. So yeah, you just take the gut sample, you extract DNA out of it. In this case, we run PCR, so we're amplifying a certain part of a gene. In this case, it's the 16S rRNA gene. If you've ever heard of that, you might, because that's what I'm going to study. So that is just a portion of a gene within bacteria that's very conserved. All of these bacteria have it, but they have slight variations, so you can tell what the different bacteria are. Yeah.
Oh, of course, it's all the way the other side. I'm coming.
Great talk. So we've done a lot of work in Glenwood Canyon adjacent to Hanging Lake looking for stem tetrapods, and these shells are everywhere because they get super bleached and they're super easy to find. We never find the actual snails though, just what they've left behind. Why would the snails leave Hanging Lake and go up to these super rocky, barren cliff faces? What are they doing up there?
That's a really good question. I'm not so sure that they're leaving Hanging Lake. I think that, well, I don't know after the fires. The fires may have driven them out or maybe killed them off. It's a really good question. We're not sure what drives where they decide to be. There's a lot of species that are so endemic to just like one particular mountaintop that they're their own subspecies of snail. And so they can be really specific to their habitat. We're not sure what makes some of them want to live under cliff faces and what makes some of them want to live in like Hanging Lake. I would want to live in Hanging Lake if I was a snail. But they're not dependent on water. So even though I said they're at Hanging Lake, they're not aquatic snails. They live kind of at the base of trees and things like that. So yeah, if you go into the forest, if you go into these montane habitats, it's really easy to spot the dead shells. Let's see, where's a picture of this snail? So if you imagine this shell, actually that's kind of an example there. They just become very sun bleached, so they naturally have that kind of brown color. When the snail dies and when it becomes desiccated, it turns into this really white sun-bleached color. That's all of the calcium in the shell. So then they become very easy to spot. But if you find the white shells, that usually means that there will be live snails somewhere. So if you see them, you should look a little bit closer, harder, and really try. They camouflage really well into their environment, but once you find one, you can easily find a whole lot of them.
Other questions? All right, thanks, Bridget, very much.
Thank you.